All right, today we're going to tackle a really tough topic: biliary tract cancers. Now, these are a group of aggressive malignancies that have been notoriously difficult to treat. For a long time, they were kind of a black box in oncology. But now, some pretty groundbreaking research is finally starting to crack them open, showing us what makes them tick and, most importantly, how we might be able to stop them. So, what's the big problem we're up against? Well, biliary tract cancers, or BTCs, have a really tough prognosis. For anyone with advanced disease, the standard chemo options are pretty limited, and doctors have been searching for new, more effective ways to fight back for a long, long time. BTC isn't actually just one single disease. It's more like a family of cancers that pop up in different parts of the biliary system. So depending on where they start, they get different names. You've got intrahepatic, which is inside the liver, extrahepatic, in the bile ducts outside the liver, and then gallbladder cancer. 3 very different locations, which means 3 distinct subtypes. And this, right here, is the heart of the problem. Even though they start in different places, for decades, these 3 cancers were all just lumped together and treated with a one-size-fits-all approach. And that approach was almost always chemotherapy, which unfortunately just didn't work very well. It was kind of like trying to use the exact same key to open 3 completely different locks. This all leads to the really powerful question that sparked the research we're looking at today. What if treating them all as one big thing was the fundamental mistake? What if, when you look at them on a genetic level, you're actually facing 3 completely different enemies, each with its own unique blueprint? So to get some answers, scientists launched this massive molecular investigation. They were determined to crack the genetic code of these tumors, to find out if they really were different. And the scale of what they did was, well, it was seriously impressive. And when I say impressive scale, I mean it. Researchers analyzed the molecular profiles of 1,502 different biliary tract tumors. This wasn't just a quick peek. This was a deep, comprehensive dive into the entire genetic landscape. They basically threw a whole arsenal of advanced techniques at it. Think of them like molecular detectives using every tool they have. They used next-generation sequencing to actually read the tumor's DNA, immunohistochemistry to look at its proteins, and a bunch of other methods to analyze RNA. All of it was a hunt for any clue, any pattern that could tell these cancers apart. And what they found? Oh, it was a total game changer. The biggest takeaway from this huge study was crystal clear. Biliary tract cancers are not one disease. At the molecular level, they are, without a doubt, 3 separate diseases. Okay, now this is where you can really see that discovery in action. Each set of bars you see here is a different gene, and the colors stand for our 3 cancer subtypes. So just for example, look at the gene labeled IDH1. See that huge green bar? That's the intrahepatic cancers. The blue and red bars are almost flat. This is the visual proof right here. They're not just a little different, they have completely different genetic signatures. So let's just break that down a bit. Each subtype has its own unique molecular fingerprint. The intrahepatic ones, IHCCs, they're often driven by mutations in genes like IDH1. The extrahepatic ones, EHCCs, you see a lot more KRAS mutations. And then with gallbladder cancers, GBCs, you often find way too much of a protein called HER2/neu. These are totally distinct biological drivers. And this is where it starts to get really, really exciting. Because, identifying these differences isn't just for academic papers. If you can understand your enemy's unique blueprint, you can start to find its specific weak spots. This slide shows us exactly how that works. The researchers took all those genetic fingerprints and started grouping them into what are called therapeutic pathways, which is just a fancy way of saying pathways that we might already have drugs for. So for instance, on the left, you see things like homologous repair deficiency. These aren't just random mutations anymore. They've suddenly become actionable targets. Let's dig into a key example. One vulnerability they found is something called homologous recombination repair deficiency. Basically, it's a flaw in the cancer cell's own DNA repair kit. And they found this specific defect in an incredible 19.1% of gallbladder cancers. That's a huge therapeutic opportunity right there. So how do you take advantage of a weakness like that? Well, with a class of drugs called PARP inhibitors. These drugs are literally designed to attack cancers that have this exact DNA repair problem. So you've got a cancer with a specific weakness and a drug that targets that exact weakness. It's a perfect match. And this kind of profiling opens up opportunities everywhere. Take immunotherapy, for example, one of the most powerful new tools we have in oncology. This analysis found that 11.4% of all these biliary tract tumors had at least one biomarker, which is kind of like a biological signpost that predicts they'll respond well to immunotherapy. But, and this is the theme, right? It's not one size fits all. As you can see here, the potential for immunotherapy is way higher in the IHCC and GBC subtypes, around 12 to 13%. But for EHCC, it's only about 7%. This just hammers home the point that you have to test each tumor to figure out which treatment path holds the most promise. So what all this data points to is a completely new roadmap for patients and their doctors. It really lays out a whole new way of thinking about how we should approach these cancers from day one. What this research really does is transform biliary tract cancer from a disease with very few options into what oncologists call a target-rich disease. I mean, just look at this chart of protein expression. For a protein like HER2, over 9% of some tumors have it. For another one called c-Met, it's over 40%. Every single one of those is a potential target with a potential therapy to go after it. The study's own conclusion really says it all perfectly. We have to start using molecular profiling in the clinic. We need to analyze every single tumor to guide which therapies or even which clinical trials are the right fit for that specific patient. The era of just guessing is over. So the big takeaway for everyone, doctors, patients, families, is this really clear three-step path forward. First, you diagnose the specific subtype. Second, you profile its unique genetic code. And third, you targeted specific weakness with a precisely matched therapy. It's just a much smarter and much more hopeful way forward. Ultimately, this research lets us reframe the entire conversation. We get to move away from a broad, often ineffective question and instead ask a highly specific and much more powerful one. The future of treating these really tough cancers isn't about finding a stronger poison. It's about crafting a smarter key, one that's designed to unlock and dismantle one specific tumor at a time.